Have you ever seen people with difficulty walking? It's one of the symptoms of autosomal dominant hereditary spastic paraplegia, in short, AD-HSP. A neurodegenerative disease in which patients suffer from leg spasticity and difficulty walking. The current available drug only targets spastic symptoms, for example, oral antispastic drugs and baclofen pump. Hazan et al. Shows that the most common cause of AD-HSP is due to mutation of spastin protein. Spastin is known to regulate microtubule growth by severing microtubules. The Spas protein code is found to be 49% identical between humans and Drosophila, making Drosophila a wonderful model to study AD-HSP. When AD-HSP is modeled in Drosophila via loss of spastin function, adults demonstrate weak locomotor activity, while the larvae actually shows bunch motor neuron morphology, as we can see here, and even weaker synaptic signals. Our lab's previous research discovered that suppression of P21 activating kinase, PAC3, in cells that express gliotactin potentially rescues the spastin loss morphology. Gliotactin is widely found in subperitoneal glio, implicating a role for this glial cell in spastin mutant pathogenesis. To understand whether the loss of PAC3 in subperitoneal glial is sufficient to rescue spastin mutant synaptic phenotype. In this research, I utilized another subperitoneal glial specific promoter from the Moody gene to drive the suppression of PAC3. I first carried out several genetic processes to build my desired phenotype. For example, Wildtype, spastin mutant, PAC3 RNAi and spastin mutant, PAC3 RNAi expressed in the expression of Moody gene. So PAC3 expression is knocked down in all subperitoneal glial. And again, PAC3 RNAi expressed in the expression of Moody and spastin mutant. I then dissect the third instar larvae of all these genotypes and image them and quantify their terminal boutons under a compound fluorescence microscope. In the wild-type Drosophila, I observed an average of about 6 to 7 terminal boutons, while in spastin mutant, I observed twice as many, an average of 12 to 13 terminal boutons. However, when I suppress PAC3 in subperitoneal glial, in a spastin mutant, observe a dramatic rescue of spastin mutant phenotype, which looks exactly like the wild type, with an average of 4 to 5 synaptic bouton. My result confirmed that PAC3 is in subperitoneal glial and it regulates abnormal synaptic growth in spastin mutant. So why is this happening? PAC3 is known to regulate the formation of actin filaments in subperitoneal glial. Subperitoneal glial are known to act like astrocytes in which actively surveys the neuronal environment through its extensive projections. In spastin mutant, I suspect that subperitoneal glial actively interact with neurons that suffer from spastin mutation, resulting in formation of bunch neuronal morphology. However, without PAC3 and subperitoneal glial, there will be no actin form or less actin form, resulting in the lack of interaction between subperitoneal glial and neurons. Therefore, there's no bunch morphology. To understand the synaptic growth occurs under the influence of subperitoneal glial, I plan to conduct live cell imaging on third instar larvae to visualize the interactions between neurons and subperitoneal glial across time. Ultimately, through understanding the cellular interactions between all these players while comparing them to wild types, we will be able to design more specific drugs to target specific cellular players, thereby creating a healthier future for all these patients. I want to thank my PI, Dr. Nina Sherwood, Dr. Emily, for guiding me through this whole process. Also, thank you, Duke and Duke Kunshan Universities for sponsoring this research.